So today we will continue from the tissue. Last class we have started the tissue. Already different type of tissue we have completed. The first, tissue is divided into two types, plant tissue. Number one is meristematic. Second is permanent. Second tissue is permanent tissue. Permanent. Already meristematic part we have completed last class. Different type of meristematic tissue and what are the questions, important questions already last class I had told you. If you have any doubt still in the meristematic part, you can ask in your comment section. Okay, so I will clarify at the end. So next is permanent tissue. If you come to the permanent tissue, already definition we have discussed last class. Those who have lost the capacity of division. The permanent tissue is made of that cells who lost the capacity of division, means they cannot produce new cells. The number of cells cannot increase now in the permanent tissue. And permanent tissue are categorized into two groups. Permanent tissue are of two types. Permanent tissue is categorized into two types. One is a simple permanent tissue. Another is called complex permanent tissue. So first come to simple permanent tissue. What is simple permanent tissue? Its name indicates simple. This simple permanent tissue are made of only one kind of cell, one kind. Diagrammatically I am showing here. Closely look at the diagram. If a permanent tissue is made of only one kind of cells, that is called simple. But what is complex permanent tissue? If a tissue is made of two or more kind of cells, you can see in the diagram, two or more kind of cells, that is called as a complex permanent tissue. And here different type of simple permanent tissue are there. One by one we will discuss. First, simple permanent tissue are three types. Number one is called parenchyma. These names you have to remember. Parenchyma. Second permanent tissue is called as a collenchyma. And third permanent tissue is called as a sclerenchyma. Parenchyma, collenchyma and sclerenchyma. Now we will not discuss about meristematic tissue. Meristematic tissue already you have completed in the last class. Okay. So parenchyma, collenchyma and sclerenchyma, all the three different types of tissue are made of only one kind of cells. So all the three comes under this simple permanent tissue. But if you come to the complex permanent tissue is made of two or more kind of cells. And main example of complex permanent tissue are mainly conducting tissue. What is the meaning of conducting tissue? Inside the plant body, those tissue which helps in conduction of water and food. Just like in case of animal body, there is conduction of blood conduction takes place by arteries and vein. Similarly, in case of plant body, the complex permanent tissue are known as conducting tissue because they helps in conduction of water and food. And what is the conducting tissue in case of plant body? Number one is called a xylem, which helps in conduction of water. All of you know from the lower classes, the xylem helps in conduction of water and minerals. And second, the phloem helps in conduction of food. So xylem and phloem comes under this complex permanent tissue. So important questions what are asked in the NEET. So first, if you come to the simple permanent tissue, all the three simple permanent tissue, their character differs. So that's why there are three different types. Simple permanent tissue, their definition is the same. All are made of one type of cells. But in case of parenchyma, collenchyma and sclerenchyma, you find few differences. What are the differences? First, the parenchyma and collenchyma, these are the living tissue. These are the living complex tissue. Whereas the last one, sclerenchyma, this is the dead. This one is the dead simple permanent tissue. First two are living, but the sclerenchyma is dead. And another important difference, if you come to the first one, parenchyma constitute the major component of plant body. Major component means maximum part of the plant body is made of this parenchymatous tissue. So parenchyma forms the major component of plant body. Sometimes question comes, major component of plant body. So if question comes, major component Major component means major part of plant body is made of which tissue? That is parenchyma. And a second important characteristic of collenchyma, what is the important characteristic of collenchyma? This collenchyma is not found in monocot, it is only found in dicot. This collenchyma, if you come to the characteristic of collenchyma, both are living, parenchyma and collenchyma are living, but collenchyma only present in dicot, present in dicot plant, not in monocot. Collenchyma is only found in dicot, and it is found in young stem and leaf. It is found in the leaf, petiole part of leaf. Leaf means petiole part. What is the petiole of leaf? Petiole means the part of leaf which helps in attachment to the stem. This is the leaf part. The leaf attached to the stem by a stalk. This stalk is called as a petiole. So this part contains this collenchyma. Collenchyma is present in the petiole of leaf, and just below the epidermis. Last class I had told you what is epidermis and hypodermis. If you look, the outer covering of plant, outer covering is called as epidermis, and just below the epidermis different layers are present. Outer covering is epidermis, then hypodermis, cortex, endodermis, pericycle, central path is pith. So different circles, concentric circles, last class I had told you. So first is epidermis, second is hypodermis. If you look at this hypodermis part, this part, the hypodermis generally contain collenchyma. So sometime question comes, which part of dicot plant contain the collenchyma? Collenchyma is present just below the epidermis, means in hypodermis, present in dicot. And in dicot, which part? It is present in hypodermis. The collenchyma is present in hypodermis part. But in case of monocot, the same hypodermis is present, but it is not made of collenchyma, it is made of sclerenchyma. So hypodermis of dicot is made of collenchyma, but hypodermis of monocot plant. In case of monocot plant, single cotyledon plants, in case of monocot plant you find sclerenchyma. That is the difference you have to remember. And only sclerenchyma is the only dead tissue, but first two, parenchyma and collenchyma, are living. So now question comes, why the sclerenchyma is dead? Initially when they are born, when they are divided, they are living, but the sclerenchyma become dead in the latter stage, and this death of the sclerenchyma become dead due to deposition of a chemical in their cell wall. All of you know the plant cell is covered by cell wall. I am showing the sclerenchyma, one sclerenchyma cell. This is the sclerenchyma cell. This sclerenchyma, generally if you look at the cell wall, this is a cell and this is the wall portion. The wall of the sclerenchyma is deposited with a chemical known as a lignin. You have to remember. So that's why the wall is called as a lignified wall. What is that wall? It is called a lignified wall. You have to remember. Lignified wall. Lignified wall means deposited with lignin. If a cell wall is deposited with lignin, if the cell wall is deposited with lignin, that is called as a lignified cell wall. And due to this lignified cell wall, the wall become very hard and it is impermeable to different substances like water and nutrient. So water and nutrient cannot easily enter due to deposition of lignin. Cellulose is a different thing. Through the cellulose, water and different nutrients can enter, but this lignin make the wall impermeable. So that's why nutrient cannot enter. So that's why the cell become dead. There is no protoplast, there is no protoplasm, no cytoplasm, no nucleus. Everything is absent inside. Only it is a cover. It is a wall, lignified wall. So that's why you can say generally dead cells. If you look at the dead cells are generally contain this lignified wall. Whatever the dead cell you find inside the plant body, mainly it is deposited with lignin or suberin. Maximum you find lignified. So that's why remember, when the dead cell will come, there is a deposition of lignin, lignified, and other are living. If there is no lignin, only cellulose is present. So that is parenchyma and collenchyma, that is living. So we have completed this simple permanent tissue completed. Now come to the complex, because maximum question comes from your complex permanent tissue, that is xylem and phloem. So now one by one we will discuss about xylem and phloem. Perhaps this simple permanent tissue is clear. So now you can take a screenshot of this, you can write at the end after completion of class to make your notes. So now we will discuss about xylem and phloem in detail, because maximum question is asked from this xylem and phloem. So first, xylem is a complex permanent tissue. So about simple permanent tissue now we have completed. So perhaps there is no doubt in the simple permanent tissue. If still you have doubt, you can ask in the comment section. So now come to the complex permanent tissue, that is xylem and phloem. Xylem and phloem. So first, xylem. Xylem, other name, xylem is also called as a hardrome and phloem is also called as a leptome. So first come to xylem. Sometimes these questions can be asked. Number one is xylem. Xylem is also called as a hardrome. Hardrome and phloem is called as a leptome. So in this chapter we will not discuss about what is the function of xylem. Here we will discuss about what are the structural aspect, what is the anatomy of xylem. Anatomy means internal structure. Already last class I had told you. This xylem, as it is a complex permanent tissue, it is made of 4 kind of cells. Already the definition I had told you. If it is made of 2 or more kind of cells, that is called as a complex tissue. So now come to the xylem. What are the cells present in xylem? Xylem is made of 4 kind of cells, 4 types of cells. How many types of cells are present? 4 types. So one by one we will discuss. Number 1 is called as a tracheids. Number 1 is known as a tracheids. Tracheids. Second are known as a vessels. Third is known as a xylem parenchyma. And last is called as a xylem fiber. Number 4 is xylem fiber. So 4 kind of cells which make the xylem. So that's why xylem and phloem are the complex permanent tissue. First question comes in neat about these 4 kind of cells. Out of 4 kind of cells, 3 cells are dead and only 1 is living. Can you guess which one is living? I had already told you. So what are the living simple permanent tissue? Simple permanent tissue, 3 types I had told you. One is parenchyma, collenchyma, sclerenchyma, and one parenchyma term is written here. Parenchyma, if you see, tracheids, vessels, xylem, parenchyma. So already I had told you, parenchyma and collenchyma are living, and parenchyma is a major component of plant. So that's why we can say xylem parenchyma is the living. So this one, xylem parenchyma is the only living cell. Only living cell present in case of xylem, rest are dead. Xylem fiber is dead, and these 2 also, tracheids and vessels, these are also dead. Tracheid is dead and vessel is dead. So that's why you have to remember only one is living. Nothing to remember here. Only remember one thing, that is xylem parenchyma is living, rest are dead. So clear, first point is clear. So new student, if you are facing difficulty, there is no problem. Directly you listen what I am explaining. So previous videos you can refer. Okay, in your app it is given. So there is no problem. All the previous videos are available for you. There is no problem. Don't worry about that. So but listen what I am explaining today. So tracheids, vessels and fibers, these are dead, and only xylem parenchyma is living. So that's why we can say, if question will come, why these cells are dead? Tracheid is dead, vessel is dead, and xylem fiber is dead. Already I had told you, if a cell is dead, their wall is deposited with which chemical? Lignin. So lignified wall. Next. So next character is lignified. First character is living or dead. Second character we will discuss the lignified wall. Lignified wall present or absent. Lignified wall. If question will come, lignified wall present in tracheid. Yes, lignified wall present. Lignified wall present in vessel. Yes, present, because it is dead. But lignified wall is not present in case of xylem parenchyma. No. So xylem fiber, again the lignified wall is present, and inside the wall the living part is called as a protoplast. If question comes protoplast present, third point, protoplast protoplast. Protoplast means protoplasm. Protoplasm and cell membrane. Protoplast present or not? Protoplast present. As the first one is dead, protoplasm present, no, protoplast is absent because it is dead. Again, vessel, protoplasm is absent, no, protoplast is absent. Living, protoplast is present, yes, because it is living. And this one, again dead, so protoplast is absent. So protoplast is only present in living, you have to remember, because protoplast is the living part. Protoplast is the living part and lignified wall is the dead. So if cell is dead, then lignified wall is present. If cell is alive, then protoplast is present. Clear? So 3 points we have discussed. Which one is living, which one is dead, which one having lignified wall, and which one having protoplast. Now one by one we will discuss the shape of the cell. Here only few characteristics we have discussed. Now we will go to the shape of the cell and their function, the main function and the shape of the cell. Now this part is clear. Now you can take a screenshot of this. Okay, so I cannot repeat these things, so you have to take the screenshot with the interval when I am explaining. Okay, so this character is completed. 3 characters we have completed. Which one is dead, lignified wall, and which contain protoplast. Now after this we will discuss the shape, because sometime the question will come from the shape of the cells found in the xylem and phloem. So now come to the shape of the cells. First come to the tracheids. Generally if you look at the tracheids cell, these tracheids are generally elongated with tapering ends. Elongated means long. You can see their length is more. Generally a cell is a sphere. In case of animal you have observed, generally this cell is a sphere, 3-dimensional structure, sphere, or it is a rectangular or polygonal having many sides. But if you look at the tracheid, what is the importance of tracheid? If you look at the tracheid, first it is elongated. What is the character? First, elongated. Elongated means long, length is more. Elongated. This cell is elongated. And the ends are tapered. Ends are tapered means pointed. You can see this end, you can see this end. The end are pointed, not blunt. This end are pointed. So this is called as a tapering end. Tapering end. This end also comes as a question. In case of tracheids, which type of end are found? This is a tapering end. And as it is a dead, their wall is deposited. The wall is deposited with what? Already I had explained you. This deposited with, this wall is deposited with your lignified wall. So this is the lignified wall. This wall is deposited with lignin. The wall is deposited with lignin. This is the lignified wall, because all the dead cells is deposited with lignin. So this is the lignified wall. Lignified wall deposited with lignified wall. Clear? So this is the lignified wall. And one important thing you have to remember, this lignin which is deposited, it varies from tracheid to tracheid. Here I have shown only one tracheid, but inside plant body you find millions of tracheid, thousands of tracheid. And if you look at the tracheid, different tracheid, one tracheid having more thickness of lignin or lignified wall is more thick. In some tracheids the lignified wall is thin, so it varies in thickness. The lignin deposition varies from tracheids to tracheids. So this is all about your tracheid. So next come to vessels. If you come to the vessel, next important cell is vessel. If you compare the characteristic of tracheids with vessels, generally vessels are cylindrical. If you look at the vessel is generally cylindrical. You can see this is a cylinder. Cylindrical. It is not having tapering end. This is the wall of vessels. And generally vessels are multicellular. Multicellular means one vessel connected to another vessel. Again that vessel is connected to another vessel. Again its upper side connected to another vessel. So vessels form a tube, long tube. So vessel, what is vessel? Vessel is a long tube present inside the plant body for conduction of water, because the xylem helps in conduction of water. What is the important thing? If you compare the end In case of tracheids, the end is pointed, but in case of vessels, the end is not pointed, and vessels are multicellular. Tracheids are unicellular. Tracheids are unicellular. Unicellular means made of one cell. Here it is a single cell, but vessel is a long tube and it is made of many cells. And each cell is called as a vessel element or vessel member. This is one, this is second, third, fourth, each one. This is a vessel member, this is a vessel member, this is a vessel member, this is a vessel member. So one vessel member is connected to another vessel member end to end, end to end. The end of first vessel member is connected to second vessel member. The end of second vessel member connected to third, like that. So it is called vessel element. Vessel element. So vessel element is unicellular. Vessel element is unicellular. Vessel element, single element is unicellular, but complete vessel is a multicellular. And if you look at the end, this vessel end, this end are generally transverse, or it is a plate-like structure. It is not a tapered, it is a pointed, but it is not pointed. It is having a plate-like structure. And this, if you look at this end, this is called end plate. And the end plate having pores or perforations. There is holes. In the end, there is holes, and these holes are called as perforations or pore. Perforation, vessel pore. Vessel pore. These are called as a vessel pore. And that's why this is called as a perforation plate. This plate is known as a perforation plate. What is the name of this plate? It is called as a perforation. Perforation. Perforation means a hole bearing plate. Perforation plate or end plate. And what is the function of this perforation plate? Due to perforation, due to presence of hole, this hole is present in everywhere. What is the function of these holes or pores? These pores helps in conduction of water, because when root absorbs water from the soil, root absorbs water, water moves in upward direction from one vessel element to other vessel element, like this. From one vessel element to other vessel element, water can be transported in upward direction. Clear? So that's why this is perforated. So what is the main difference between tracheid and a vessel? Here there is no perforation. In case of tracheid there is no pore, it is only pointed. But in this case the end is perforated. The ends are having pores. Clear? So that's why vessel is the main water conducting element. So maximum water inside the plant body is conducted by these vessels. And if you compare, out of these four cells I had told you, tracheids, vessels, xylem, parenchyma, and last is xylem fiber. Out of these four, only these two, the first two, tracheids and vessels, helps in conduction of water, and the last two provide support. The first two only helps in conduction of water, so that's why these are called as a tracheary elements. Tracheary elements. Sometimes question comes. Tracheary elements. Tracheary elements is made of. What are the tracheary elements? Tracheary elements is made of two type of cell. That is tracheid and vessels. Another important characteristic. Out of this tracheary element, tracheid and vessel, this vessel is not found in all plants. Vessel is the only characteristic of angiosperm, present in angiosperm. You have to remember, this question is asked many times in NEET. Present in angiosperms. Angiosperm, different type of plant are there. Thallophyta, Bryophyta, Pteridophyta, Gymnosperm, Angiosperm. It is only present in angiosperm, means absent in gymnosperm. In case of naked seeded or gymnosperm, those seeds are visible in plant. So these plants in which the seeds are visible, these are called as a gymnosperm. In case of a gymnosperm, the vessel is absent, only present in angiosperm. You have to remember, this angiosperm contain only vessel. So vessel is the only characteristic of angiosperm without any exception. Clear? So tracheids, vessels completed, tracheary elements. But these two provide support. Xylem parenchyma is living. It stores only food. The living, this xylem parenchyma, it synthesizes some amount of food, and it also helps in storage of food. The food is stored. So that's why xylem parenchyma and xylem fiber, they are mainly supportive tissue, supportive cells. So that's why we have to not discuss about their shape and function. So if you want to remember the shape, xylem fiber are just like tracheids. They are also tracheids, elongated, and some fibers are like spherical like this. And in case of xylem parenchyma, shape varies. Xylem parenchyma, they are isodiametric, generally spherical cells, spherical or polygonal, sometime like this. But if one shape, it continues for all. If spherical, all the xylem parenchyma are spherical. If it is rectangular, all the xylem parenchyma are rectangular. So like this you can remember, but you have to remember first about these two tracheary elements. Tracheary elements, clear? Tracheids and vessels, characteristic of tracheids and vessels are important, you have to remember. And one important thing, last class also I had told you, there is a two type of tissue according to development of plant body. When a plant is in early stage, it contains the primary tissue, and when a plant become adult, that contains secondary tissue. So that's why xylem are two types, one is a primary xylem, another is a secondary xylem. So now you have completed this part, all the tissues completed. Now I am going to remove these two because these two are not important, xylem parenchyma and fiber. Okay, so if you come to the xylem are two types again. Xylem tissue are two types, one is primary xylem, another is called as a secondary xylem. Already I had told you what is the difference between primary and secondary with an example in the last class. If still you have not remembered, I am telling again that example. Primary school, you have read in primary school, LKG, UKG up to class 7, standard 7, primary school. After 7, 8, 9, 10, that is your secondary school. Now you are in higher secondary. Similarly, in case of plant body, there is two type of tissue. When a tissue is produced in the early life, young, when a plant is young, that is called as a primary xylem. And when tissue is produced in later stage, that is called as a secondary xylem. And the secondary xylem are called as a wood. Secondary xylem is called as a wood. What are the wood you are using? Different wood of the plant you are using for furniture in your home. Different furnitures are made. The woody substances which are collected from the plant body, that is nothing the secondary xylem. So secondary xylem's other name is called woods, but primary xylem is never called as woods. So you have to remember, primary xylem are not that much strong, so that's why primary xylem are not called as a wood, you have to remember, clear? But the primary xylem are again two types. One is called protoxylem. Primary xylem are two types, protoxylem, another is called as a metaxylem. So we are not discussing about secondary xylem later, but those xylem which are produced in early in plant life, so primary xylem are of two types. What are the primary xylem? One is a protoxylem, another is a metaxylem. What is the difference between proto and meta? Sometimes question comes. Proto and meta, those primary xylem which are produced in the first when the life of the plant starts, that is called as a protoxylem. But later, those which are produced, these are called as a metaxylem. It is not understood. Okay, so again I am telling the same thing. So what is the difference between protoxylem and metaxylem? First I had told you primary and secondary. Primary means when a plant is young, when a plant is old, secondary. But now tell me, I have given you one example. What is the example? Primary school and secondary school. When you are in primary school, from which class you have started? Which class, LKG or UKG? That is called proto, because starting, that is the starting, fundamental. So similarly, primary xylem, no doubt, primary xylem is found in young. But in that young stage, again which comes first, that is called proto, and after proto those comes, that is called meta. Means your UKG or LKG, that is proto, and class 1, 2, 3, that is called as a meta. Just like protozoa you have read, all of you read protozoa. Proto means first, zoa means animal, first animals, protozoa. Similarly, protoxylem, first xylem which comes in plant life, that is called as a protoxylem. Similarly, after protoxylem those come which are called as metaxylem. Okay, So after to listen carefully with maximum concentration, because I am going to ask you question relating to this portion. So what I am explained here, I will ask the question and you have to answer in the comment section. I will see the answer of each and every student. Okay. So protoxylem and metaxylem. By using this protoxylem and metaxylem, we will discuss one question. Because important part is this protoxylem and metaxylem. Last class I had told you the different layers of stem and root. If it is a plant body, this is a plant body, this is the soil surface, below you find root, above you find shoot. This is the stem portion. Upper part of the plant is stem, lower part of the plant is called as root. So if you cut a plant transversely, if you cut a plant like this, what you find? You find circular layers. Circular layers. First is called epidermis, then hypodermis, cortex, endodermis, pericycle, center is pith. Center is pith. Yes or no? So these layers I had told you already in the last class. So first is called epidermis. Epidermis, second layer is called as a hypodermis, third layer is called as cortex. Cortex or general cortex, then is endodermis, endodermis, then pericycle, pericycle central part is called as a pith, and in between pith there is a xylem and phloem. Xylem, phloem are present. So that's what we have discussed, and already I had told you how to remember these layers. एक हाई क्लास इंग्लिश पेपर ऑलरेडी आई हैड टोल्ड यू, एक हाई क्लास इंग्लिश पेपर एपिडर्मिस, एक एपिडर्मिस हाई, हाइपोडर्मिस क्लास, कॉर्टेक्स इंग्लिश, एंडोडर्मिस पेपर, पेरिसाइकल. तो दीज आर द लेयर्स फ्रॉम एक्सटर्नल टू इंटरनल, आउटर टू इनर. Already in the last class, first class, I had already told you so about these layers. But important thing is this xylem. Xylem is present between pericycle and the center pith. In between these two there is a xylem and phloem. But the arrangement of xylem and phloem is different in stem and root. If you look at the stem, you find circular layers like this. In case of root also, if you cut, you find circular layers like this. But difference is the arrangement of protoxylem and metaxylem. So now we'll discuss what is the difference with respect to root and stem. Root and stem. So now tell me, what is the center of the plant? Center, exactly at the center what is present? Central part is called as a pith. And already I had told you xylem, phloem are present between pith and pericycle. So if you look at the stem portion, what is found in case of stem? In case of stem, if you look at the protoxylem, the protoxylem is found towards center, means pith. Protoxylem is present towards pith. And metaxylem, those which are metaxylem means after protoxylem. Those which are new xylem means after protoxylem are produced, they are present towards pericycle, means periphery. So only you have to remember, in case of stem, the protoxylem is at a center, means towards pith. Question comes, in case of stem, protoxylem is present towards pith or center. Here we will not use center word. The center is a mathematical term, and pith, the answer is your pith. It is close to the pith. Pith is the center of the plant. Clear? So it is protoxylem is in stem. Protoxylem is present towards pith, and metaxylem is present towards periphery or pericycle, we can say. But opposite is seen in case of root. In case of root, you find opposite. The metaxylem is at a center, but protoxylem is in the periphery. So protoxylem, PX means protoxylem. Protoxylem is present towards periphery, metaxylem is present towards center. So by this arrangement, we can differentiate root and stem. Sometime question comes. Already this question is asked in NEET. This question is already asked in the NEET. What was the question? How you differentiate, how you recognize a root and a stem, how you differentiate a root and a stem. Let two parts given to you. One stem is given to you and root is given to you, and they are very old. From outside you cannot distinguish them because it is a section. After cutting it is given, a slice is given. How you find whether it is a root or stem? If you look at the protoxylem, metaxylem, it is just opposite. Protoxylem is at pith. In case of stem, protoxylem is at periphery. In case of root. So now tell me, first, in the first case, in case of stem, protoxylem is present in the center, means the inner side. When anything present inside, it is called as endo. All of you know endo word is used, just like endosmosis. You have read in chemistry endosmosis, exosmosis. When osmosis takes place and water enters inside cell, that is called as endosmosis. And when water comes outside of the cell, from the cell it comes to outside, that is called as exosmosis. So similarly here one endo and exo word is used. In case of a first stem, the protoxylem is at center. Center means it is present inside the metaxylem. Protoxylem is present inside the metaxylem. So that's why this condition is called as end-dark condition. So very important, you have to remember. So maximum time this question is already asked. What is end-dark? Two type of question can come. First question come, in case of stem, protoxylem is present towards which part? Pith. As protoxylem is present towards pith, center, it is present in the inner. It is present inside the metaxylem. Metaxylem is outside, but protoxylem is inside. We are discussing proto. Proto means first. With respect to first, we are writing the definition. It is called end-dark. Similarly, what will the condition here? You tell in case of root. In case of root, as protoxylem is outside. Outside means out means exosmosis. As protoxylem is out, it is called as exarch. These two conditions you have to remember, and here nothing you cannot want. Here in this case you cannot just remember, you have to visualize these things. In case of end-dark and exarch, what is happening directly you cannot remember these things, so you have to visualize the concept. So when inside protoxylem is present, that is end, just like endosmosis, inside protoxylem end-dark and outside protoxylem, that is exarch. Clear? So this is all about your end-dark and exarch condition in case of stem and root. Clear? And already these questions are asked, and this completes your xylem completed. But now I am going to ask you one question what is already asked in the NEET, and it is very important, frequently repeated, because in biology new questions are not there. In biology questions are fixed, and these questions are repeated every year. So that's why if you solve the last 5 or 10 year questions, 60% question you get repeated directly in the NEET biology. So now I am going to ask one question. Already you have written or taken the screenshot. Now we'll move forward. So what is the question? Already I have told you trachea element. The question is about the trachea elements. Trachea elements means tracheids. So listen the question carefully. So what is the question? Tracheids differ from other trachea elements. Other trachea elements. Tracheids differ from other trachea elements in which of the following condition? In which of the following condition? Following condition. The question is clear. Tracheids differ from other trachea element in which of the following condition? Because tracheids number one option have lignified wall. Second option, without nucleus. Third, tracheids are being incorporated because tracheids are being imperforated. Fourth, all of the above. Just 15 seconds I am giving, you have to answer because already you have read. So you have to answer in comment section. Okay, I will check the answer of all the students. So tracheids, first listen the question. Tracheids differ from other tracheary element. Tracheids differ from other tracheary elements. What are the tracheary element? Tracheary element means tracheids and vessels together. Tracheid and vessel both are together called as a tracheary element. And question is asked, tracheids differ from. What is the question? Tracheid differ from. Tracheids differ from other tracheary element means how tracheid different from vessels. How tracheid different from vessel means which character is present in tracheid but absent in vessel, which make the tracheid different. Because tracheid possesses, so means from out of these four options you have to choose that option which is present in only tracheid, not in vessel. The first option, lignified wall. If you want lignified wall must be present only in tracheid. If lignified wall is the answer, then it must be present only in tracheid, not in vessel. So you have to choose that option which is only found in tracheid, because we have to differentiate here how tracheids are different from vessel. Okay, now you have understood the question. Now I am explaining the answer, then I will check what you have written. So first tell me, the option 1 is written lignified wall. Lignified wall is found in case of dead cells. Already I had told you. And out of these two cells, tracheids and vessels, both are dead. Already I had told you. Tracheids, vessels and fiber are dead. Only parenchyma is living. So first one is not the answer. It is the common character. It is found in tracheid. Tracheid have lignified wall, vessels have lignified wall. And if a cell is dead, protoplast is absent and nucleus is also absent, because nucleus is a part of protoplast. So nucleus is absent in both. As both are dead, so dead means nucleus absent. And third option is written being imperforated. Being imperforated. Perforated means pore is present. Imperforated means pore is absent. If you look at the tracheid and vessel diagram, already I had told you, the tracheids are like this with tapering end and vessels are like this, and vessels possesses pores. So vessels are perforated, but these are imperforated. You never find pores. So that's why answer is being imperforated. The tracheids are imperforated. All is not the answer. So if only one is answer, these two are not the answer. Why all of this? So being imperforated. Being imperforated. Okay. So tracheids are imperforated. You have to remember, tracheids are imperforated and vessels are perforated. This question is already asked in 2014 NEET and repeated many times. After that, so xylem we have completed. If you have any doubt from this xylem portion, you can ask. After this xylem, we will discuss about the phloem part. What is phloem? Students, phloem is the conducting tissue which helps in conduction of food and organic food, mainly organic foods and some minerals. Enzyme hormones are also transported. So next is phloem. Phloem. Phloem is also called as a leptome. Xylem is also called as a hardrome. Phloem is called as a leptome. And this phloem is made of four kind of cells. Again, just like xylem. Phloem is made of, first is sieve tube. Sieve tube. Second is known as companion cell. Companion cell. Third is called as a phloem parenchyma. Phloem parenchyma, and last is phloem fiber. Fourth one is phloem fiber. One by one we'll discuss. So first, already I had told you, first character you have to know which are living, which are dead here. In case of xylem I had told you only one is living. Which one is living? Only your xylem parenchyma is living, rest are dead. But in case of phloem, just reverse. In case of phloem, you have to remember, only one is dead, three are living. So if question comes only one is dead, which one is dead? You guess. Just like living, parenchyma is always living. Dead means which one is dead? Sclerenchyma is dead. And already I had told you the fibers are modified sclerenchyma. Fibers are a type of sclerenchyma. So that's why only fiber is dead. You have to remember. In case of a phloem, only fiber is dead, but these three are living. Sieve tube is living. Companion cell is living. Phloem parenchyma is living. Parenchyma is living from the beginning. It will never be dead. So phloem fiber are dead. You have to remember. Only one is dead. So that's why if question comes protoplast absent or nucleus absent, protoplast absent in case of fiber, protoplast absent, I can write protoplast absent, nucleus is also absent. Nucleus at the center of the cell there is a nucleus. Protoplast absent as well as a nucleus absent. But in rest 3, in case of sieve tube, companion cell and phloem parenchyma, there is a presence of protoplast and a presence of nucleus with only one exception which is frequently asked. In case of all the living, if a cell is living, protoplast is sure. If a cell is dead, protoplast absent. But one important thing, there is an exception in case of sieve tube. You have to remember. Sieve tube is living, but it lack nucleus. Nucleus absent. Cytoplasm present, but nucleus absent. Nucleus is absent. You have to remember. In case of sieve tube, the nucleus is absent, but rest material except nucleus, cytoplasm and some organelles are present in case of sieve tube. So that's why due to absence of nucleus, some functions are absent in sieve tube. Sieve tube cannot perform 100% function inside the phloem. The sieve tube cannot perform all of its function because sieve tube is incomplete. There is a nucleus absent. No doubt it is living, but nucleus is absent, and it can be comparable with one cell of animal. In case of animal body, in case of mammals mainly, not animal, in case of mammal I can say. In case of mammal, one cell lack nucleus. All of you know that is human RBC or mammalian RBC. In case of RBC, nucleus is absent, but still it can live for 120 days inside our blood. The lifespan of RBC is 120 days. So just like RBC, the sieve tube is living, but nucleus is absent. So due to absence of nucleus, what happens? Due to absence of nucleus, in case of sieve tube, some functions of sieve tube cannot be performed. So that's why in case of sieve tube, one cell helps, that is called as a companion cell. Companion means which gives company. So that's why it is called companion cell. So companion cell performs some function of sieve tube. Some function of sieve tube is controlled by companion cell. So if question comes, nucleus is absent in case of sieve tube, it is true. But due to absence of nucleus, some functions are absent. These functions are controlled by companion cell. So companion cell perform some of the functions of sieve tube. And what are these functions? The companion cell maintains a pressure gradient inside the sieve tube. Companion cell, what is the function of companion cell in case of sieve tube? The companion cell maintain pressure gradient. Pressure gradient means pressure difference. Pressure gradient in sieve tube. In case of sieve tube, the pressure gradient of sieve tube is controlled by companion cell. Direct question comes. In sieve tube, pressure gradient means pressure difference. The pressure difference of sieve tube is controlled by companion cell. The nucleus of companion cell, because nucleus is absent in case of sieve tube. Okay, but the nucleus is present in these two, in case of companion cell and parenchyma. So now we will discuss their structure, because sometimes direct from the structure of the cell question comes, because we have discussed for xylem also. So that's why now we have to discuss for phloem. Now come to the structural aspects of the cells of phloem. First is the sieve tube. If you look at the sieve tube, it is a tube-like structure, just like in case of xylem we have discussed, the vessels are tube-like. Here in case of phloem, you find there is a tube-like structure. There is a tube-like structure. In case of phloem, that is called as a sieve tube. Sieve tube. So this is the sieve tube, and already characteristic of sieve tube we have discussed. Sieve tube is multicellular. Sieve tube is a long tube, just like vessel, it is a multicellular, and each cell is called as a sieve tube element. Sieve tube, each single one, the single one is called as a sieve tube element. Each single one is called as a sieve tube element. And if you look at the sieve tube element, there is no nucleus, but cytoplasm is present. Cytoplasm is very thin, it is present towards periphery. And important thing, if you look at the end wall, it is not pointed. The end wall is flat or oblique, it can be bent also. But important thing, you find pores, openings. One sieve tube is connected to another sieve tube. One sieve tube is connected to another sieve tube, just like one vessel is connected to another vessel by perforation, pores. These are called as a sieve pore. These pores are known as a sieve pore. What is the opening? This pore is known as a sieve pore. This pore or opening is called as a sieve pore. Sieve pore. And this plate, this complete plate, end plate, this plate is called as a sieve plate. Sieve plate. Sieve plate. This plate is called as a sieve plate. And just adjacent to sieve tube, there is another cell, elongated cell like this you find, with almost pointed end. This is called as a companion cell. Two companion cells together I am showing. This is one companion cell, this is the nucleus. So you can easily compare. This is the companion cell. Companion cell. This is the sieve tube, this is the companion cell. The function of sieve tube and a companion cell, they regulated because the nucleus is present in companion cell and the nucleus is absent in case of sieve tube. Now question comes, in case of sieve tube and companion cell, their wall is present. This is the wall of companion cell, and this is the wall of sieve tube. How the nucleus of companion cell perform some function like maintaining pressure gradient in case of sieve tube? There is no connection. But if you look carefully, this wall, the wall of sieve tube and the wall of companion cell, at some places they are very thin. Very thin. I am showing the thin portions. If you look at this point, in this point you can easily compare. This is the wall of sieve tube. This is the wall of the sieve tube, but you find here this is a thin portion. Similarly, this side, this is the wall of the companion cell. This is the thick wall, thick portion, this is the thin portion. And these thin portions are called as a pit or pit fields. Pit. And pit are the thin area through which different substance from one cell can be connected with other. The cytoplasm of one cell connected to other cell because of thin area. So that's why some chemicals present in this cell can be transported to this cell through the thin area, not through this thick area. So that's why nucleus of a companion cell can regulate the pressure gradient. How it maintains pressure gradient? Inside plant body, all of you know sucrose is a chemical, sucrose. During photosynthesis, first sucrose is synthesized, and sucrose is a disaccharide. These are the sucrose. And after formation of sucrose, first it comes to this companion cell. First it is produced in the leaf cell. All of you know photosynthesis takes place in the leaf cell. From the leaf it comes to this cell, companion cell. Then from companion cell it enters into sieve tube. And when it enters into sieve tube, the amount of sucrose increases. When amount of sucrose increases, to balance the sucrose, more water enters. More water enters from this cell as well as from xylem. This is a phloem. From the xylem contain water. From the xylem also water enters. So when more and more water will enter into more and more water will enter into this sieve tube, what will happen? The pressure will increase at this portion. There is high pressure. In this part there is a high pressure, but at opposite end there is a low pressure. So due to high pressure at one point, low pressure at other point, there is a difference in pressure, that is called as a pressure gradient. So companion cell maintains pressure gradient. Companion cell maintains pressure gradient in the sieve tube. So sieve tube cannot maintain pressure itself, pressure gradient. The pressure gradient of sieve tube is maintained by companion cell. So sieve tube and companion cell we have completed. Now sieve tube and companion cell completed. Both are living, but the function of sieve tube is controlled by companion cell. Next come to phloem parenchyma. This parenchyma is living, and one important thing about phloem parenchyma, it is absent in monocot. Absent in monocot. It is only present in dicot. Absent in monocot. Two things we have discussed. In case of simple parenchyma, simple permanent tissue, I had told you three simple permanent tissue are there: parenchyma, collenchyma, sclerenchyma. The collenchyma is only found in dicot. It is absent in monocot. Similarly, the parenchyma, phloem parenchyma, not all parenchyma, phloem parenchyma is absent in monocot. It is only found in the dicot. And fiber is present in all, and fiber is dead. And these cells only provide support. Phloem parenchyma is present here like this. Similarly, phloem fibers, these are present here. But these two cells cannot be shown. So only important first two cells, that is sieve tube and companion cell. But important thing you have to remember here, the sieve tube and companion cell only present in angiosperm, just like in xylem I had told you. In case of xylem, what I had told you, in case of xylem, the vessel is only present in case of angiosperm, and vessel is absent in gymnosperm. Similarly, the first two, sieve tube and companion cell, are completely absent in gymnosperm. In case of xylem, second one is absent. In case of xylem, second one is absent, that is vessel absent in gymnosperm. But if you compare the phloem, in case of gymnosperm, what is about gymnosperms? In case of gymnosperm, the sieve tube is absent and companion cell, both are absent. But instead of sieve tube and companion cell, two other things are present. You have to remember, in case of gymnosperm, there is a sieve cell, not sieve tube. There is a single structure, tube-like structure is absent in gymnosperm. This tube is absent. Single cells are present, sieve cell. So instead of sieve tube, what is present in gymnosperm? Sieve cell. And in case of companion cell, there is albuminous cell. Albuminous cell. These two cells you have to remember. So gymnosperm lack sieve tube and companion cell, but contains sieve cell and albuminous cell. This is all about your sieve tube and companion cell, phloem parenchyma, and last is phloem fiber. One important thing I am going to tell about this phloem fiber. Phloem fiber is also called as a bast fiber. Phloem fiber is also called as a bast fiber because of their thickness or hardness. These are called as a bast fiber. And these phloem fiber or bast fibers are used for rope making. The long ropes are prepared. So that's why these are commercially important. Question comes, which fiber is commercially important? Commercially, we can market it commercially. The phloem fibers or bast fibers are commercially important. Why they are commercially important? Because by using this bast fiber or phloem fiber, we can make the ropes just like cotton rope. We can make the rope just like example. Some examples comes. One is a jute plant. All of you know jute plant is used for rope making. Another is flax. Another is hemp. So jute, flax, hemp, these plants, bast fiber or phloem fiber. The phloem fiber of these plants are collected and used for making rope. So commercially these are used. So commercially important. Phloem fibers or bast fibers are found in case of jute, flax, hemp. Sometimes question comes, in case of jute, which fiber is used? Xylem fiber or phloem fiber? Answer is phloem fiber. Xylem fiber is never used commercially because xylem fiber only they can form a wood, but they cannot commercially used. They cannot be used for rope making. So phloem fiber or bast fiber are very important, and xylem fiber are not that much important. Xylem fiber are very less in amount in case of xylem. So that's why we have completed now the phloem part, xylem and phloem completed. So the conducting tissue or the complex permanent tissue completed. If you have any doubt, you can ask in the comment section about the phloem part. Okay, otherwise we'll go forward. This much for today's class. If you have any doubt, this much for today's class. If you have any doubt, you can ask in the comment section. Next class we will discuss about the doubt portion and what are the questions I had told you. That is important. You have to practice, and you have given you will be given with some assignment in few days after completion of this class. So you have to solve that assignment, and we will discuss one by one these questions, important questions for NEET. Thanks to all of you.